Good afternoon. I would like to thank the organizers for the invitation and thank you for your time. I will be speaking about Stevens-Johnson's and TEN, or severe adverse drug reactions in oncology patients related to anti-cancer drugs. Some of my disclosures. As a matter of background, cancer is a prevalent disease. It is the second most common cause of death worldwide. Over 14 million cases of cancer, excluding non-melanoma skin cancer, are diagnosed every year, leading to about 8.2 million deaths worldwide. Slightly increased proportion in men than in women, and in this country, 1 out of 2 men and 1 out of 3 women will be diagnosed with cancer at some point in their lives. This map illustrates the regions with the darker blue with a higher incidence of cancer, usually associated with tobacco, obesity, and diabetes, as well as a longer life expectancy. In oncology patients, which is the world that I have lived in during my entire career, polypharmacy is very common. About 2/3 of all cancer patients will receive some type of systemic chemotherapy, targeted therapy, or immunotherapy. More than 70% will receive agents for associated symptoms such as infections, pain, or seizures. And about 50% of all cancer patients will receive radiation at some point during their therapy to mitigate or their disease. In oncology, the language we use is based on the common terminology criteria for adverse events, which was initially developed by the NCI and is currently mandated in every clinical trial. This adverse events criteria states that all adverse events, including dermatologic toxicities such as rash, are graded from 1 to 5, 1 being the mild and 5 being death related to that adverse event. This is of great importance in terms of dermatologic adverse events, as any rash that is grade 3 or worse dictates immediate interruption of the anti-cancer therapy, which could be life-prolonging or life-saving. Grade 3 toxicities, including rash, are characterized by being medically significant and covering more than 30% of body surface area. We conducted an analysis from the Cancer Therapeutics Evaluation Program in a prospective fashion for 10 years and found that in over 3,500 patients, grade 3 or worse dermatologic adverse events were seeing in about 2.4% of patients, most of which occurred during the first 3 cycles of therapy. And this has important implications not only in patients' quality of life, health, but also in terms of clinical outcome, as grade 3 toxicities, as stated previously, will limit the dose of the drug that can be given, and the maximum tolerated dose, as defined as the dose that will not lead to unacceptable toxicities, is usually the one that is identified during the phase 1 trials and will ultimately be used in the marketing of the drug. A study retrospective that was conducted in Rome in over 10 years in 30 cases of Stevens-Johnson's and TEN found that the most common comorbidity in these patients was cancer, but that in all cases of Stevens-Johnson's TEN, the condition was not attributed to an anti-cancer therapy, but rather to a coincident medication, usually antibiotics or NSAIDs, and that the mortality of these cancer patients compared to the non-cancer controls was not different. We sought to investigate which agents may be culprits in Stevens-Johnson's and TEN that had been published in the literature. So we investigated from the initiation of Ovid and PubMed up until 2013, a study of those agents that were most common culprits. And as you can see here, we found agents such as lenalidomide, thalidomide, as well as other agents used mostly in hematologic malignancies were associated with Stevens-Johnson's and TEN in a total of 83 reports. All of these agents were subsequently reported to the FDA's Adverse Events Reporting System. But in the FDA's Adverse Event Reporting System, we also, through the Freedom of Information Act, sought to investigate which agents were being reported as a culprit for Stevens-Johnson's and TEN. And there were over 3,650 reports also implicating agents used for hematologic malignancies. Interestingly, of all of those agents that have been reported to the adverse events reporting system, only 2 had also been published in PubMed. So there appears to be a greater reporting to the FDA and not as much reporting in the published literature. What has changed things recently has been the introduction of novel agents targeting cancer characterized by agents that block inhibitory molecules in the immune system. Monoclonal antibodies, such as the antibody ipilimumab blocking CTLA-4, which is a negative regulator of T-cell activity, and PD-1 for programmed Death-1, also being blocked by a monoclonal antibody, are able to unleash a patient's immune system, thereby attacking cancer and, in many cases, reverting to a controlled or benign phenotype, leading to this class of therapies as being named by the journal Science in 2015 as the greatest breakthrough in cancer therapy. As you know, the first agent was approved in 2011, but subsequent to that, a number of other agents targeting similar pathways or proteins have been approved for both solid and hematologic malignancies. And subsequent to this, a number of reports have also ensued in patients who have developed a maculopapular rash initially that has responded to topical steroids, but upon subsequent administration of these immunotherapies has progressed into a case of Stevens-Johnson's and TEN. Another case with the PD-1 inhibitor nivolumab showed that a patient who presented with a rash, which is frequently seen in about a third of these patients, a maculopapular rash, was initially biopsied, showing no significant alterations, and the PD-L1 expression in skin was not significant. The patient continued to receive nivolumab after combined therapy with ipilimumab, and the patient progressed into a case of TEN, as you can see here, characterized with the typical histologic changes on H&E, but also with upregulation of PD ligand 1 in the epidermis. In addition to this, a number of cases were being reported in patients who initially had received immunotherapies. One could expect their immune system to be activated, and they progressed in terms of their cancer, so were then placed on the BRAF inhibitor vemurafenib, which is associated with a maculopapular rash in about 40% of cases. And what was found was that a series of cases in which patients received immunotherapies and then vemurafenib, developing cases of TEN seen here, with abundant CD8 positive T-cell infiltrate. This is a series of cases at our institution of 13 patients treated with ipilimumab, which progressed in terms of their cancer, were then placed on vemurafenib. This was reported in the New England Journal in 2012. And as you can see, of these 13 patients, a greater number than expected, 4 developed a grade 3 rash, as you can see here, which is much greater than anticipated, likely occurring due to an already activated immune system. Interestingly, in all of these patients, interruption of therapy with the vemurafenib, administration of oral corticosteroids, and then rechallenging with vemurafenib did not lead to a recurrence of this severe maculopapular rash. And since in oncology, we don't have the luxury of switching medications frequently as one could do with antibiotics or other medications, in most cases, we have to rechallenge patients with the culprit of their maculopapular rashes, even if they are severe. And we found that the recurrence of these rashes is not always the case, and patients can continue on therapy despite having a severe toxicity. And the interest in rashes to immunotherapies has only grown, as seen here, with a number of publications on rash to nivolumab, ipilimumab, or atezolizumab, or pembrolizumab, which are the currently approved agents. What has also caught the attention of the dermatology community is that a rash that occurs in these patients is not typical of the characteristic maculopapular rash, which is the most common. It appears like in about 10% of patients, the rash has a more lichenoid appearance, as seen here, affecting all parts of the body, including the mouth. Histology is typical of sporadic occurring lichen planus, as seen here, with an abundant lichenoid infiltrate in the dermis with CD4 and CD8 positive T cells. And to make things a little bit more confusing, it turns out that some of these agents can also cause blistering disorders. And there have been a number of reports, and from a prospective study, it was estimated that in 2.4%, bullous pemphigoid with serum positivity for anti-BP180 and 230 antibodies have been reported in patients, as seen here. Interestingly, these patients respond very well to topical corticosteroids. But a more recent paper published in Clinical Cancer Research, which I thought deserved significant attention, is the comparison of patients that develop Stevens-Johnson syndrome and lichenoid rash to immunotherapies. As you can see here, the corresponding histology, and also the staining for PD-1 and PD-ligand 1, which were the targets of these immunotherapies, were more significantly upregulated in Stevens-Johnson's, perhaps an attempt of the skin to tamper down on that inflammation and that immune attack that has been triggered by the immunotherapies, not present as significantly in the lichenoid rash, but yet still positive. But what was interesting was that when gene expression was analyzed, the lichenoid rash from immunotherapies targeting PD-1 was not similar to a maculopapular drug rash in controls, but was very similar in terms of certain genes being expressed in Stevens-Johnson's TEN, suggesting that perhaps in some cases, the lichenoid rash to immunotherapies and this immune activity is perhaps a subclinical or a form first of Stevens-Johnson, in which there is a direct attack of the patient's immune cells, especially CD8 positive and CD4 positive T cells on the skin, leading to varying degrees of expression in terms of the phenotype. In addition, what has also motivated the study of these rashes in these patients is that it turns out that with all of these immunotherapies, patients who develop worse cutaneous toxicities appear to live longer, perhaps indicating a more robust immune system that is available for the toxicity in terms of the maculopapular or the lichenoid rash, but also against the tumor. It also supports the notion that these patients who develop these toxicities are the ones who could benefit the most from them in terms of their anti-tumor effect, so we need to make every effort possible to maintain them on therapy. So we see many of these patients at our institution that develop these toxicities. As you can see here, we have analyzed some of them. Dr. Azael Freitas is presenting a poster at this meeting, and what he's done is he's analyzed 21 patients that have developed with these toxicities and looked at certain serum markers. And what we have done here is that, taking from our oncology colleagues the premise of not looking at skin biopsies or not looking at tumor biopsies, but looking at biomarkers in sera, which are much easier to obtain than skin or tumor biopsies, we found that a variety of markers, including IL-6, are upregulated in patients that develop these toxicities, as well as interleukin-6, which appear to correlate with the improvement at the subsequent visit when compared to baseline. Interestingly, patients who develop the maculopapular rash with significant pruritus to immunotherapies also appear to have elevated levels of IgE. We expect this difference to be of further significance with increasing number of patients, as seen here in 21 patients. And the same occurs for IL-6, which has been described before as being associated with severe toxicities. So based on these observations, I think there is an enormous opportunity to target in a different way the underlying mechanisms of toxicities occurring to anti-cancer therapies and potentially Stevens-Johnson's and TEN. We know that JAK inhibitors are currently approved for use in polycythemia vera, myelofibrosis, and psoriasis. These are potent agents that block the JAK kinase, that is a single transductor for the activation of T cells. Ruxolitinib and tofacitinib have been shown in vitro to downregulate interferon gamma, perforin, and granzyme B. And these effects have been taken advantage of by transplant physicians, and currently cases of steroid refractory GVHD have been treated with JAK inhibitors, such as ruxolitinib, with striking responses, and currently there's at least a dozen clinical trials ongoing with JAK inhibitors in GVHD. The same as was mentioned previously with IL-6, as you can see here, we know that it is upregulated in Stevens-Johnson's and TEN. There is an antibody that is already approved for use and has shown benefit in other conditions such as SLE and urticarial vasculitis, tocilizumab. And finally, I would like to conclude on a positive note that in some cases, patients who develop Stevens-Johnson's and TEN, again, perhaps due to that more robust immune response, their tumor appears to respond better and they are disease-free from their malignancy after their Stevens-Johnson's and TEN. So to conclude, immunotherapies may result in greater severity of a maculopapular rash caused by another drug or an atypical presentation of an SJS-TEN. Our key translational finding would be to use serum markers as predictors and prognosticators of SJS and TEN as opposed to skin biopsies, and that specific inhibition of CD8 positive T cells and IL-6 with already approved agents provides a novel opportunity for therapies, and we believe that these toxicities will increase in importance as these drugs enter earlier in the stages of cancer and they are used in combination, and of course, patients are living longer. Thank you very much.
Thank you. Dr. Wenheng Chung from Changgung Memorial Hospital in Taipei.
Thank you for giving me the chance to give the talk about how to get started? Okay. So give a talk about the update in biomarker and the therapeutic target. So although we have no many specific actually linked to specific drug-induced Stevens-Johnson syndrome or drug hypersensitivity, there's still one mystery in this past we are not clear that how the danger signal can result in very unique clinical pathological feature in Stevens-Johnson syndrome. That is extensive epithelial necrosis with fewer inflammatory cells compared to other drug hypersensitivity phenotype. So this is an historical characteristic of delayed drug hypersensitivity. For Stevens-Johnson syndrome and TEN, the major immune cell are CD8 positive T cell or NK cell compared to other less CD4 T cell, other cell. But the major cytokine was cytotoxic protein in Stevens-Johnson, guanylation, phospholic, and is more predominantly expressed in the skin region or the cytotoxic T cell. In previous, we have identified the guanylation as the major key cytotoxic protein that can trigger is a key protein that can result in the extensive keratinocyte death in Stevens-Johnson and TEN. And when you measure the blister cell, blister free in the patient skin, we can detect 100 or 1,000 times higher level of guanylation spray in the skin compared to other cytotoxic proteins such like phospholic and granzyme B and perforin. The guanylation is expressed majorly by the CD8 cytotoxic T cell or CD8 CT56 NK cell. In addition to the guanylation, that the major cytotoxic protein involved in Stevens-Johnson syndrome, there are also many cytokine or chemokine being found involved in the immune pathogenesis, such like IL-6, IL-15, IL-2 cytokine. So I would like to make a summary of how the immune mechanism involved in the Stevens-Johnson syndrome TN when the drug recognized by the T cell and then the activated T cell immigrating into the skin lesion, they can spread several cytokines or cytotoxic proteins such like guanylation or phospholic or granzyme B perforin. And for guanylation, they have a dual function. The one function is trigger the keratinocyte death. The other function, the guanylation can act as an element that can stimulate the dendritic cell or macrophage to spray more chemokines such like CCR3 and CCL20 that can attract more inflammatory cells to the virtuous cycle in the skin lesion. That can explain why the fewer inflammatory cells can induce so extensive keratinocyte death. For cell death mechanism involved in Stevens-Johnson syndrome and TN, in addition to phospholic and guanylation death signal, there are also other new findings. that have been identified, such like iron signal and also the adenosine A1, the signal interact with the formula peptide receptor 1 that can trigger the another apoptosis necroptosis, and also some specific microRNA being involved in the cell death signal in Stevens-Jones syndrome TN. So after we know the guanylate as a very specific status protein is present in Stevens-Jones syndrome TN, we take the advantage to develop our correct with Dr. Abe. We develop a rapid diagnostic kit that can use in very quickly. We can detect the distinguish Stevens-Jones syndrome or status T-cell-mediated bullous dermatosis can detect within 5 minutes. We can get the positive result. That can very easily distinguish from other autoimmune mediated bullous disorder such as pemphigus or pemphigoid, and also some chemotherapy or target therapy induced a harmful skin reaction that could be similar to Stevens-Jones syndrome. So later is different. So we have developed such kind of very easy diagnostic kit. So another benefit from this kind of rapid diagnostic kit is we can monitor the clinical course during the patient progress to TN. So for some ambiguous conditions, such like a patient with recurrent blister lesion developed in the later stage of TN, we need to use the skin biopsy to prove this is a reactivation of a saturated T cell, or this could be another bullous condition such as ischemia-induced bullous lesion. So for this example, the patient in the TN in the later stage developed new bullous lesion. We use the quick diagnosis and show the negative for the guanylate expression, and that is consistent with our skin biopsy result, showing that is thrombosis vessel with ischemia change that related to DIC, not reactivation of Stevens-Jones syndrome. So, are there any other cytokine or chemokine could be a good prognosis marker that can predict the prognosis of Stevens-Jones or TN that can correlate with the conventional score 10 factor? So we have done our cytokine study. We collect the serum sample from patient in acute stage from Regiscam member from Taiwan, Germany, and France. So we collected more than 155 cases of serum sample from patient with Stevens-Jones in acute stage. So we indeed identify some cytokine or chemokines that are specifically expressed in the serum, specifically higher than other non-Stevens-Jones syndrome drug hypersensitivity, such like trace or macropapillary rush. The cytokine we identify IL-6, IL-8, and the guanylate and IL-15. They are specifically higher expressed in the serum of Stevens-Jones or TN. And we also further evaluate whether this kind of cytokine could be correlated with the severity and prognosis. So we evaluate the score 10 of this kind of this patient and correlate to the cytokine we identify, and only IL-15 and guanylate, they are correlated with the severity and as well as the score 10 factor. So as we know, the IL-15 have the function that can activate the NK cell or cytotoxic T cell, as well as can enhance the killing effect to the target cell. So this is example some patients, they even after 1 week or 10 days from the onset of the disease can still progress to a very severe and progress to very extensive skin detachment. So some patients can detect the very high level IL-15 as well as guanylate in even 1 week or 10 days in the later stage. So after we know the pathomechanism of Stevens-Jones syndrome TN, one thing we want to get is whether we can improve our treatment. So the IVIG since have no functional role that involved in the pathogenesis, but the corticosteroid could be benefit, but it's general inhibit all kind of immune system. So we are trying to find out some specific drug can inhibit to the T-cell or cytotoxic T-cell to express guanylacine more specifically. So until now, the treatment protocol, there are no consensus specific treatment for Stevens-Jones syndrome and TEN unless a supportive care. So by understanding the immune mechanism, so we can use the ex vivo model or in vitro model that can get the T cell from the patient or get the beta cell from the skin lesion. So we can use this kind of activity, SSS-specific T cell, then can screen potential drug such like IVIG, cyclosporine to prove the concept before clinical trial. So here is the example of our ex vivo model for the potential screening of drug can use for the Stevens-Jones syndrome TN. So if we compare to the corticosteroid, the biologic anti-TNF-alpha inhibitor shows a similar inhibitor effect to the guanylacine expression by the SZTN T cell, blister cell. So because there have been several reports, there's a separate treatment for the Stevens-Jones syndrome by the biologic anti-TNF-alpha. So we have done the randomized control study prospectively. We collect 48 patients treated by entanercept, that's a biological anti-TNF-alpha blocker, and compared to the corticosteroid arm, there was 45 cases enrolled. And the result shown here, because the sample size is still not big, so we use the previous way we evaluate predict mortality by score 10 factor. So the predict mortality for the entanercept group is about 20.8, and the observed mortality is 8.3. That is more than 50% reduction of than predict mortality. And in the corticosteroid group, the predicted mortality is still about 20, but the observed mortality 15. So it's pretty much similar. So, but when we compare the direct to entanercept group to the corticosteroid group, there are no statistical significance, but it could be due to the limited sample size. But for the other, in addition to the endpoint of mortality, we also evaluate the skin improvement comparing entanercept to the corticosteroid, and we found that the entanercept group that have a significantly shorter duration for complete skin healing compared to the corticosteroid group. And another benefit for the entanercept group is have significantly lower side effect of gastrointestinal hemorrhage compared to the corticosteroid group. And because it's not ethical to enroll the third arm of supportive care, so we compare the two groups to our retrospect data that in the same medical system in the same clinical trial period. So we have more than 30 cases they are supportive care without using any IVIG or immunosuppression. So we compare the mortality. The mortality for the supportive retrospective supportive group is around 20%, more than 20%, but compared to the entanercept group is less than 10%, so that is a significant difference. So what is the underlying therapeutic mechanism around the corticosteroid and the entanercept? We have found that both corticosteroid and entanercept have significant effect to decrease the expression level of guanylacine and also the TNF-alpha level by this in the basal cell or the serum level of guanylacine or TNF-alpha. And also another benefit, we found the entanercept can upregulate the Treg, the cell population of regular T cell, but not by the systemic corticosteroid. So we all concerned the what is anti-TNF-alpha treatment. That is the tragedy of a thalidomide that can is supposedly can inhibit the TNF-alpha action, but when the thalidomide increase the mortality instead of decrease the mortality. So in addition, try to explain the reason why the thalidomide that increase the mortality. We done the in vitro study to show when the thalidomide compared to the biological anti-TNF-alpha, biological anti-TNF didn't increase the the cytotoxic effect to the keratinocyte. But the salidroma, in addition to inhibit the T cell function, has also the cytotoxic effect that is add-on cytotoxic effect to the keratinocyte. So I think it could be the reason the salidroma can increase the mortality compared to the biologic agent anti-TNF-alpha. We now developing ideally to developing the more specific anti-guanine monoclonal antibody to neutralize the antibody, the cytotoxic protein. But unfortunately there are only few big pharma. They are interested for this kind of orphan disease. So we are trying very hard by ourselves to develop this drug. But very lucky, some drug companies, they have developed some drug that can also have potential use to inhibit the guanine. So we collaborate with some drug companies that give some drug, such like a TOL 101. This drug can specifically inhibit the alpha-beta T cell receptors that can target in the T cell activation and without additional side effects. So have it showing to have successful treatment for the patient with kidney transplantation. We have done the ex vivo evaluation for this drug, and we found this drug is very sufficiently inhibit the guanine expression of the Stevens Johnson T cell, blister cell. That's pretty much similar to the efficacy compared to the corticosteroid. So we have other ongoing clinical trials because the limit of sample size, so we need to have multiple center, multiple country correlation. So in Asian country, we have our SCAR group joined with different Asian countries. Also, the RIGI SCAR group from the French, Italy also joined the clinical trial. So hopefully in the future we have more and more a result to show alternative treatment in addition to corticosteroid, and we can find out more treatment to improve outcome of Stevens-Johnson syndrome. Thank you for your attention.
It is a great pleasure to have an opportunity to present our recent results. I'm from the Niigata, Japan. Niigata is so famous for the delicious rice and sake. Okay, first I would like to introduce my previous study results. I follow the Wilfans paper, early diagnostic marker of SGSTM, granulation, serum granulation level is the early diagnostic marker of SGSTN. And also, as Wilfan introduced, we developed a rapid test for granulation. So it's so useful to distinguish the early stage of SGSTN and also the MPE or EM. In addition, we recently developed SGSTN model mouse using SGSTN patient sample, PBMC. So we injected this PBMC to the immunocompromised mouse, and following causative drug administration orally. So as you can see, this model mouse showed hemorrhage in the eye. And recently, we found the keratinocyte death mechanism in SGSTN. In addition to apoptosis, we found necroptosis also occurred in the SGSTN skin. And today, I will show the other topics. This is a photo of Stevens-Johnson syndrome case. Maybe no doubt this patient is a Stevens-Johnson syndrome, but as discussed in this meeting, it is so difficult to identify the cause. This is a drug or viral or mycoplasma. For example, if this patient has a pneumonia and treated with antibiotics, we usually diagnose this patient is a Stevens-Johnson syndrome caused by the antibiotics, but it's true. So I would like to talk about Helicobacter pylori, not virus or mycoplasma. As everybody you know, Helicobacter pylori colonizes the gastric mucosa of at least half of the human population, and also Helicobacter pylori infection represents a key factor in the etiology of various diseases such as chronic active gastritis and gastric adenocarcinoma. So recently, we treated this Helicobacter pylori. Standard triple treatment includes a proton pump inhibitor, clarithromycin, and amoxicillin, administered for 7 days. But we usually, we frequently see the patient with skin rash associated with Helicobacter pylori eradication therapy. And we noticed these patients show so unique character phenotype. For example, this skin rash appears 3 days after treatment finish. It means 3 days without no medication. And also, to identify the cotabit drug, we usually examine LTT, lymphocyte transformation test, but 3 medication shows all negative, and also, honestly, we are so very careful, so we repeated 3 times, but all tests show negative. So from this experience, we consider that this skin rash is not caused by drugs. Helicobacter pylori is involved in the mechanism of skin rash. So to address this question, we perform this assay, measurement of cytokines from patient PBMC exposed to Helicobacter pylori. We collect the PBMC from the patient. So, and we kill the Helicobacter pylori, then that Helicobacter pylori were added to this PBMC. Then we collect the supernatant. Then we measure the cytokine in this supernatant. If there are some immune reaction against the Helicobacter pylori, some cytokine should be released from this PBMC. This is the result. As you can see, only HP plus SR plus, it means the patient with skin rash show the high concentration of IL-6, IL-2, whole interferon gamma, and TNF-alpha, whereas patient without skin reaction, almost no increase of this cytokine. Of course, HP minus, it means HP, Helicobacter pylori non-carrier, showed no increase. To confirm this reaction, we performed lymphocyte transformation test, ELISpot, and also CD154 expression. CD154 is a marker of antigen-specific CD4 positive T cell. As you can see, in the patient, there are significant immune reaction in the lymphocyte transformation test, and also in the ELISpot, there are numerous spots in the patient PBMC with Helicobacter pylori, and also 3.84% HCD4 positive T cell is it means it's Helicobacter pylori specific. In particular, using CD154 expression, we divided the 2 groups. One group is this after 3 days means skin rash appear after 3 days or more after the HP treatment finish. And also before 2 days means before 2 days skin rash appear. As you can see, so it clearly distinguish And after 3 days, almost all the patients have a HP-specific immune reaction. So we analyzed 15 patients whose skin rash associated with HP eradication eradication treatments. Only 3 patients have a positive for the medication, and 9 patients showed a HP-specific immune reaction. From this data, we concluded that in this patient, several patients showed have a HP-specific immune reaction. So what's the mechanism? From the previous report, inflammation of chronic arthritis requires Helicobacter pylori specific T cell, and also antigen presentation of Helicobacter pylori is occurred at the pile patch in the gut. So increase of bacterial component by the treatment triggers immune reaction to Helicobacter pylori. So we focus on the exosome. How to the Helicobacter presentation. Exosome are nanovesicle in body fluids, serum, and incorporated messenger RNA and proteins. Exosome play an important role in cell-to-cell communication. Especially, exosomes are involved in antigen presentation, and exosomes are stable in the humans, even in serum. So first, we analyzed the Helicobacter pylori peptide present in the serum exosome. This is the data of proteome analysis of serum exosome. As you can see, a patient with skin rash, serum exosome contained Helicobacter pylori peptide, whereas patient without skin rash did not have a Helicobacter pylori peptide. Of course, a non-carrier, there are no Helicobacter pylori peptide. And we confirm Helicobacter pylori containing exosome induces specific T-cell activation. We collected exosome from Helicobacter pylori exposed dendritic cell. Then we add this exosome to patient PBMC. As you can see, only Helicobacter pylori induces this specific T-cell activation. So we suggest this mechanism of skin reaction related to Helicobacter pylori treatment. Drug killed Helicobacter pylori, then this Helicobacter pylori component are absorbed through intestine, and the dendritic cells phagocyte this Helicobacter pylori component, then release exosome, which is contain the Helicobacter pylori component, then induce immunological recognition of Helicobacter pylori component, then induction of immune reaction, resulting the skin rash. So from this data, we concluded Helicobacter pylori allergy, I say allergy, exists. So in this meeting, so many speakers said, not only drug, but some virus and mycoplasma induce it's like SJS. So I would like to say, maybe bacteria has a potential to induce the skin rash. So this is the only Helicobacter pylori. Other bacteria also induce the bacteria-specific immune reaction. So we analyze we collected blood from Helicobacter pylori carrier treated with this eradication treatment, and we collected several time points. I'm sorry, this is so busy table, but it's another result of bacteria peptide in serum exosome. There are so many bacteria contained in the exosome, and interestingly, compared to the day 0 and day 10, there are some new bacteria appear in the serum exosome. So I want to say, not only Helicobacter pylori, but other bacteria can induce the bacteria-specific allergy immune reaction. So in conclusion, cutaneous adverse drug reaction could be caused by bacteria in gut, and also I would say this is the reason why exanthema frequently appear during antibiotics therapy. And this is a photo of my department. Thank you for your attention.
From the clinical observation, we can find a predominant cytotoxic T cell or NK T cell or NK cell in the blister of SJS and TN. And this data suggests that the drug-specific cytotoxic T cell may be involved in the reaction. So, how the drug can stimulate T cells in SJS-TN? We propose that the specific T cell receptor clone type may recognize the drug or metabolite. The drug or metabolite may presented by the specific HLA allele, and then the active T cell can secretory the cytotoxic protein or cytokine to disrupt the tissue. But the drug antigen is very small, so how the drug interact with the HLA and the T cell receptor? There are different models to explain how the drug could be recognized by the HLA or the T cell receptor. The models include the heptane theory or the altered peptide receptor model, or the PI concept and altered T cell receptor receptor model. In these models, we can find that cell model, they require the antigen processing pathway, but several models such as heptane concept, the drug antigen was proposed to have a covalent binding with the peptide. And some of them are MHC restriction and some are not. And such kind of reaction can cause the oligoclonal or polyclonal T cell activation. So, how to validate the different model involved in immunopathogenesis of SJS and TN? We may need to consider the internal factor or outer factor. The internal factor include the HLA T cell receptor or the drug antigen. Among these internal factor, the HLA may be the most important factor, because the genetic data have shown that the specific HLA is associated with several drug-induced SJS and the TN. And there are increasing studies show the different HLA protein possess various binding affinity to the drug. In our previous study, we found that the recombinant HLA protein, such as HB1502, has binding affinity toward the carbamazepine, epoxide or oxcarbazepine, but has no affinity to the metabolite of oxcarbazepine, such as the carbamazepine. And our cell culture data also show that there is no involvement of intracellular processing. And our data suggests that the T-cell, the HLA protein may directly present the drug to the T-cell. And our data do not support the alter peptide repertoire hypothesis. And our data also do not support the heptane concept. And we use side array mutagenesis. We identify several residues in HLA B1502 allele. They are very important for the drug binding. If we disrupt the residue 63 asparagine, then the HLA could not present the drug to the T cell. Similarly, we also find that the residue in HLB5801, they are important for the presentation of the drug antigen ospirinol to the T cell in the allopurinol scar. For example, if we disrupt the residue 97 arginine, then this HLA could not present the ospirinol to the T cell. In addition to the HLA, the drug antigen is very important for the formation of the immune synapse. And we know that some drugs, they are more notorious and more common to induce the SDS and TN. And we also can find that the drug concentration increase in the patient of SDS and TN. In our previous study, we used the GWAS association study. We identified the genetic variants of cytochrome P450, CYP2C9. The genetic variant can show genome-wide association with the phenytoin-induced SDS and TN and DRES. But you can find that there is no association between the HLA alleles and the phenytoin-induced SDS and the TN. It means that although SDS and TN they are considered as the immune disease, but the drug metabolism, the defect can associate with such kind of immune disorder. And in the patient, we also observe that the phenytoin-induced scar patient, they have a poor metabolizer. We can find they have a delayed metabolism of phenytoin. So it means that if the patient has increased concentration of the drug antigen, then they have an increased risk or increase the risk to develop the immune reaction. And we also find a similar phenomena in our allopurinol scar. We find that the ospirinol, the plasma level increased in the allopurinol scar patient. And ospirinol level also increased in the patient with renal deficiency. So if the patient have a renal deficiency, they have an increased risk of allopurinol scar. So the data suggests that the increased concentration of the drug antigen can promote the immune reaction to activate the T cell. And we also examine the T cell receptor clone type in SJS and TN. We use the next generation sequencing to study the T cell receptor clone type in the blister cell of the SJS and TN. And we can find the specific clone type in the blister cell of carbamazepine-induced SJS and TN. Compared to the tolerance control healthy donor, the TCR, the T cell receptor, they are polyclonal. But in the SJS and TN patient, in the blister cell, the CDR3 show the oligoclonal and the public usage. And we applied the single cell assay. You can find that we can identify the special paired TCR alpha and beta. And the single cell suggests that this cell, they have the specific TCR alpha and beta clone type. And they express the guanine and the granzyme B. So they are cytotoxic T cells. And this public CDR3 T cell receptor, we originally identified in the blister cell of our patient, Han Chinese with HLB 1502. But we also can find such public CDR3 T cell receptor in the PBMC of the European who had no HLB 1502. So it suggests that the T cell receptor, the specific T cell receptor, they share the same sequence, and they can, they may able to bind into the drug without the HLA restriction. And we generate the recombinant protein. We find that this specific T cell receptor can directly interact with the drug antigen. For example, this kind of recombinant protein, they can directly interact with the carbamazepine epoxide or oxcarbazepine. And we perform the transfetan expression, the specific T cell receptor. And we find that the recognition of the T cell receptor to the drug can be promoted by the antigen presenting cell with the HLB 1502. And we find that this kind of the transfetan can recognize the drug antigen. And we collaborate with David Ostroff for the computer modeling, and he will present this data later. Previously, I showed you the data of carbamazepine, SJS, and TN, and we identified the public TCR. But the public TCR cannot be found in other kind of scar. For example, we can identify the clonal expansion of the T cell receptor in the blister cell of oropinal scar, but we cannot find the public T cell receptor. And finally, I will show you some data for cytotoxic protein or cytokine specific increase in the region of the SJS and TN. Previously, we identified guanine. It is a key molecule for the epidermal crisis in SJS and TN. And we found that the guanine level is correlated with the mortality or the plasma level of osprinol in oropinal scar. In addition to guanine, we also identified other cytokines such as IL-15, is very important for the severity and the mortality of SJS and TN. Finally, we need to consider the other factor may also contribute to the SJS and TN. For example, the previous infection may explain how the T cell appear. In the model of heterologous immunity, try to explain that the previous infection can cause the diversity of T cell receptor. For example, in this figure, you can find that the T cell, the virus-specific T cell receptor may appear after the infection, and such kind of T cell receptor may closely react to the drug antigen, so can associate with the SJS and TN. So according to our better understanding for the immunopathogenesis of SJS and TN, then we can prevent or predict this disease and develop the therapeutic agents. We can apply the genetic test, but many kind of scar reaction we still don't know, or we cannot find the genetic marker. So we need to consider drug antigen. We need to consider the drug-drug interaction or the underlying disease for the underlying disease may delay the drug metabolism. And the therapeutical, and we can use the identify biomarker to develop the new therapeutic agents. Thank you for your attention.
Thank you. I'd just like to say thank you to Elizabeth and everyone involved in organising the meeting today. It's been really great to hear everyone's experiences from all over the world. And thank you for the opportunity to present on the pharmacogenomics of SJS-TN. So just to put SJS-TN in context of all adverse drug reactions, SJS is considered an off-target adverse drug reaction, and a part of the off-target drug reactions are those with, that show immunological memory, and this can be antibody-mediated memory like you see in penicillin anaphylaxis, or it can be T-cell-mediated memory that we see in a Bacavir hypersensitivity, for example, and also for SJS-TN. The breakthrough genetic finding in drug hypersensitivity research was really back in 2002 with the discovery of the association between the carriage of HLA-B5701 and the risk for the development of a Bacavir hypersensitivity syndrome. And this was really a breakthrough because for the first time it showed a clear genetic link between drug hypersensitivity and T-cell mediated mechanisms. Shortly after this, there was the publication of the link between HLA-B1502 and carbamazepine SJS in the Han Chinese, and this was shortly followed by HLA-B5801 associated with allopurinol scar, also in the Han Chinese. And really investigations into these three principal drug hypersensitivity syndromes have led the way in research into T-cell mediated adverse drug reactions since this time. So since the early 2000s, there's been many other HLA associations that have been published, both in SJS-TN and also more widely in SCAR. And what's really important when we're thinking about HLA genetics and SJS-TN or drug hypersensitivity is the population frequency of the allele in question, and also the phenotype. So, for example, it's been mentioned quite a bit today, but for carbamazepine, for example, in the Han Chinese, the drug hypersensitivity is SJS-TN, and this is associated with HLA-B1502, whereas if you look at European or Japanese populations, they don't carry the B1502 allele, and in these populations, stress is the dominant phenotype, and it's associated with the carriage of HLA-A3101. I'd like to point out also that for the majority of drugs that cause SJS-TN, there's still no known HLA alleles that are associated, and this is really the first step in understanding specific hypersensitivity syndrome or SJS development for specific drugs. So this has been mentioned a little bit in the previous talk, but there are 3 models to explain how drugs can interact with T-cell receptors to cause an immune response. So very briefly, these are the hapten model. In this model, the drug binds covalently to a self-peptide, or the drug metabolite binds to a self-peptide, and then this alteration in the peptide elicits a new TCR response. The second theory is the PI model, and in this model, drugs bind non-covalently to immune receptors, and this may be the HLA molecule itself or the TCR molecule, for example. And it's a fairly broad theory, and an adaptation of this PI model is the altered peptide repertoire model that we see in Abacavir hypersensitivity. And in this model, the drug binds within the groove the HLA antigen binding cleft, and the presence of the drug within the groove then alters the chemistry of the cleft, so subsequently the peptides that can then be bound by and presented by this HLA molecule are changed. So I'm just going to talk about Abacavir a little bit because I think it helps in our understanding of what's happening when we look at SJS-TN and HLA associations. So if you think about the HLA peptide binding groove as a series of pockets, and each of these pockets are polymorphic and they interact with the side chains of the bound peptide. And here you can see a schematic of the HLA peptide binding groove and the classic 9-mer peptide that's bound to a class 1 molecule. And the 2 key pockets are really the P2 pocket, the B pocket that anchors the P2 peptide at the N terminal, and also the F pocket that binds to the anchoring P9 peptide at the C terminal. And this is just another view of that looking from above, so here you can see the B pocket in green and the F pocket in purple. And when we look at Abacavir, it was shown through crystallization of Abacavir bound to HLA-B5701, and this was published in 2012 simultaneously by Patricia Illing and David Ostrov's groups. And what this showed was that there was only 2 positions that were very important in the binding of Abacavir to the groove of HLA-B5701. And these were specific amino acids located at position 114 and 116 that are unique to HLA-B5701 and not seen in similar alleles like B5703 or B5801. And so this just shows you how very small changes in the right position with the HLA peptide binding groove can be responsible for the development of hypersensitivity. So Abacavir hypersensitivity and SJS share several features. They both exhibit a long-lasting memory. As I've said, they're CD8 T-cell dependent responses. The development of the syndrome show fairly short latency, and they can be explained by the heterologous hypothesis that was just described in the previous talk. However, the 2 syndromes are quite unique as well, and there's lots of differences that we can't really explain yet. So when we look at Abacavir hypersensitivity, there's a single drug and a single risk allele, which is quite unusual. There's very mild skin involvement. If a rash develops, it's only a dermal disease, and really it's a systemic response. Whereas if you look at SJS-TN, we know that multiple drugs and multiple HLA can be implicated in the case of SJS-TN, and sometimes no causative drug is actually identified. And here, the skin and the mucous membranes are the major site, and you get full thickness epidermal necrosis, as I said, involvement of the mucous membranes and blistering. And granulysin is a key local mediator of keratinocyte killing in SJS-TN. Also, Abacavir patients are consistently patch test positive, so this helps in phenotyping this particular T-cell-mediated adverse drug reaction, whereas this is really inconsistent when we're looking at SJS-TEN. And finally, when we look at the TCR of PBMCs from patients that have suffered from Abacavir hypersensitivity, they're generally polyclonal, while at least we know in the case of carbamazepine in SJS, sometimes these TCRs can be restricted. So now I'm just going to talk through a way of looking at cases where you get multiple HLA alleles associated with a particular drug hypersensitivity like SJS-TEN, and how this can, how you can really deal with this and get information out of this. So if you think about the alleles that are associated with carbamazepine SJS-TEN, as I mentioned, HLA B1502 is really the best characterised allele, and this has been shown in the Han Chinese and many other Southeast Asian populations now. But there's also others from the B75 serotype that are also associated, and these are B1508, B1511 and B1521. And if you look at, use a tool called MHC cluster that clusters H alleles based on peptide binding affinities, you can actually see that these risk alleles all cluster within one node, and so their preferred peptide binding specificities are very similar when compared to other HLA B15 alleles. And what's interesting, if you look at this cluster, it also identifies HLA B1515. However, I don't think that this has been reported in the literature as being associated with carbamazepine SJS-TEN. And I think the reason for that is just got very low frequency globally of that particular allele. So I think that's quite interesting. As I mentioned, however, the B1502 is really quite frequent within the Southeast Asian population, so that's the reason why this is the major allele that's been associated with this particular SJS-TEN. You can also look at particular positions along the peptide binding groove, comparing risk and control alleles, and really find out where you may get clues about what's going on with the drug in terms of the binding groove. So here you can see, if you look at a comparison between HLA B1501 and 1502, there's 4 amino acid residues that may be important. And then if you look at all of the risk alleles together, this position 63 really comes out as quite a striking position in the peptide binding groove. And as mentioned in the previous talk, this has been shown in functional studies to be the key position that's associated with responses to carbamazepine in patients. And modelling has also previously been published that shows that this is an important position for the binding of carbamazepine to HLA, but I'm not sure if David might have an update on that in the next presentation. So we know that we're quite good with negative predictive values now. If we know the risk HLA, chances are that we're going to get negative predictive values close to 100% if we look in the right population in a well-phenotyped group of patients. But the big question going forward for pharmacogenomics and for research into SJS is the positive predictive value gap. So the question remains, why do a minority of patients with a HLA risk allele develop an immune-mediated adverse drug reaction? And what is the basis of the development of these specific phenotypes? Why do some people who carry 5801 and take allopurinol develop SJS, while others get DRESS, and other people can get more mild cutaneous reactions? So part of the answer to this is to look at drug dose and metabolism, and here I've just listed 3 of the main examples in the literature that have also been discussed a little bit today. So we know that phenytoin is metabolized predominantly by CYP2C9, and the CYP2C9-3, which is a loss of function allele, shows a significant association with phenytoin-related scar, and independently, delayed clearance of plasma phenytoin has been associated with scar as well. Similarly, 2 mutations in CYP2B6 have been associated with reduced clearance of nevirapine and also cutaneous adverse drug reactions and SJS-TEN. In contrast, the wild type haplotype, the GT haplotype, which is the wild type, is protective in this particular genetic association. And this effect is strongest in Africans, but it's also been observed in Asian populations as well. So it seems that in these drug metabolizing enzymes, we're going to see population-specific effects, just like we see in HLA associations. And finally, allopurinol is metabolized to oxypurinol by aldehyde oxidoreductase, and it's been shown that renal impairment and delayed clearance of plasma oxypurinol is the independent risk factor for allopurinol scar. Is it possible just to go forward? I've just got a couple more. Thank you. So I won't go into detail here, but I'm just going to highlight that there are 2 HLA associations that are now used in pharmacogenomic screening and have been implemented successfully, and these are 5701, use of abacavir, and also B1502, in particular Asian populations before the use of carbamazepine. And the use of, or the screening for 5801 before the use of allopurinol is sort of under assessment in terms of cost effectiveness. And these particular examples have really laid out a roadmap for what's required to go from a genetic association through to a test that can be used in the clinic. Forward, please. Okay, so this is a slide that just sort of illustrates what may be future targets in genetic research in regards to SJS-TEN. And as I've mentioned, HLA continues really to be the first step in identifying and characterising a particular drug, SJS syndrome. And we have to consider population frequency of these risk H alleles and what the risk alleles have in common when we're looking across different populations. I've also mentioned about drug dose and metabolism and drug transporters, and TCR was mentioned in the previous talk as well. There may be particular TCR sequences that are important for particular drugs, but we also have to think about which cells are expressing these TCR. Are they memory T-cell responses, for example, from tissue-resident cells or recruited effector memory T-cells? And I think characterising this is going to explain a lot about the pathogenesis of SJS-TEN. We also need to remember that there's other cells that are found in the blister fluid and in the skin during SJS-TEN, the NK cells and the NKT cells. So perhaps we need to consider how KIA is affecting their activation or inhibition, and how KIA is interacting with HLA in SJS-TEN. And I think the last piece of the puzzle is what's happening with the peptide. There may be parts of the proteasome or the peptide processing pathway that are also contain a lot of polymorphic genes that may be associated with SJS-TEN for particular drugs. So just in conclusion, HLA associated with SCAR has provided a roadmap for the assessment and implementation of pharmacological screening that can be applied to any genetic test in drug hypersensitivity. Understanding HLA population allele diversity and frequency is key to identifying risk and cost effectiveness. And currently genetic testing may be useful to predict and prevent reactions, but also just to aid diagnosis if an association has been recognised, but it's not used for full screening yet. And also to identify patients who may be at risk of developing an adverse drug reaction. Despite HLA associations, the positive predictive values remain relatively low, and many other factors contribute to the development of disease. And the PP gap, or the positive predictive value gap, presents many opportunities for the further research into the pathogenesis and mechanisms of development of SJS-TEN. So I'd just like to acknowledge my research group. I'm not sure what's happened there, but that extends from Perth to the US, and particularly thank you to Elizabeth and Simon for your leadership in our group, and thank you to all of our collaborators across the world. That's it, thanks.
So what I'd like to do today is to talk about how modeling of drug-HLA interactions is based on solved crystal structures. And I want to talk about how we have undertaken modeling a set of drugs that may bind the antigen binding cleft of HLA molecules that are associated with SJS-TEN. And I want to go a little bit deeper into carbamazepine and update people on what our thinking is. Is there a pointer?
Yeah, there's a little red button with a laser on it.
The green? Okay, thanks. And then finally, if there's time, I want to mention a project where we're interested in using the same kind of assays to test skin sensitizers for binding to HLA molecules. So, as many of you may know, a number of crystal structures have been solved of class 1 and class 2 HLA molecules. This is the crystal structure of a class 1 molecule. And we have learned some general rules about how peptides interact with HLA molecules. In the case of class 1 MHC, the peptides that bind typically have the same length if they are binding to a particular class 1 HLA allele. And they typically have positions that we call anchor residues, where side chains are oriented down away from us, making close contact, important contact with the HLA molecule, holding the peptide in place. The ends of the peptide are in pretty much of the same place in all of the solved structures that have ever been solved with peptides bound to class 1. People can describe the antigen binding cleft for class 1 as a trough with sealed ends. The ends are sealed by conserved residues that make contact with the termini of the peptide. That's why the ends are in the same place in all of the structures of peptides bound to class 1 HLA molecules. And as many of you may know, T cells utilize their T-cell antigen receptors, which are made up of variable and constant domains. And these V domains interact with the surface of the peptide HLA complex that is solvent exposed, which is zoomed in here. And you can see here up there that the CDRs, the so-called complementarity determining regions, are these loops that we've been hearing about where there are sequences associated with carbamazepine SJS-TEN. So that's just a little bit of background. In terms of the crystal structure, if you can click the video to start, that would be great. As was mentioned, there have been 4 crystal structures now that have been solved of a bacavir bound to HLA-B57. 2 by Jamie Ross-John's group and 2 by my group. So this is the electron density for a bacavir in the antigen binding cleft of HLA-B57. And as we heard in previous talks, in the case of a bacavir in HLA-B57, a bacavir is underneath the peptide. It's buried. It's not directly accessible by the T-cell antigen receptor. And you might wonder, if the drug is underneath the peptide, does the drug induce some gross conformational change in the structure of the peptide? Not really. If you look at the structure of if you look at the 4 crystal structures of a bacavir bound to HLA-B57 and compare the peptides with each other, this is what you see. And it looks pretty typical. The ends of the peptide are in the exact same place as all of the other solved crystal structures of peptides that bind class 1. And the bulging doesn't, thanks so much, Simon. And the bulging doesn't occur to a greater or lesser extent than in any other structure. And so in the case of abacavir, the drug is buried underneath the peptide. The T-cell receptor presumably contacts a surface that's made up of peptide and HLA, just like a normal, conventional T-cell receptor recognition event. What about other drugs? Can we predict, based on our knowledge of HLA sequences and HLA structures and the structures of the drugs, can we predict which drugs might bind particular HLA alleles? To answer that question, we used the crystal structure of abacavir bound to peptide and HLA to see if we could do this. We did basically a control experiment to see if we could computationally recapitulate the drug-HLA interaction. And so what we did was we took the crystal structure of abacavir bound to peptide and HLA-B57. We took away the peptide. We took away all of the ordered water molecules from the crystal structure. We took out the abacavir. We docked abacavir back into the structure, and we got a score. You ask the computer program a question, it will give you an answer. The score here is the scores are expressed in a term that's meant to predict delta G. So the more negative the number, the tighter the binding. So the way that I think about this is that it takes 7.2 kilocalories of energy to dissociate abacavir from the antigen binding cleft of HLA-B57, according to this prediction. However, the orientation of the drug in the site was not right. It was not the same as the crystal structure. So then we thought, well, maybe we need more information to do accurate modeling of small molecules to the HLA. So what we did was we put the peptide back in. We put the peptide back in, and we got dramatically better binding score, according to the computer, and the drug appeared to bind the right way if we included more information. So this tells us something. It tells us that our prediction of drug-HLA interactions could be dramatically improved with more information. Information about the peptide. So you can see here a list of drugs that have caused hypersensitivity in a manner that's been associated with specific HLA alleles. And so what we did was we used molecular docking to dock these drugs in without peptide, like I mentioned that we did for abacavir, and also with peptide. And what you can see is that the small molecules fall into two categories. One category where the small molecules give a better predicted binding score in the absence of peptide, like carbamazepine. And some drugs, like abacavir, clearly are predicted to bind better in the presence of peptide. What does this mean? The short answer is that the ones that bind better in the absence of peptide are too big to fit underneath the peptide like abacavir. And I think that can be shown here, probably. So this is nevirapine, where molecular docking was used to place the structure of nevirapine in HLA CO401. And what I'm showing you are the top 9 poses predicted by molecular docking. And what you can see is that the drug is occupying approximately the area of positions 2 and 3 of the peptide. Position 2, you remember, is the site where the side chain is an anchor that fits into the B pocket. So this is an example of how we do the modeling without peptide. And this is an example of how we do the modeling with the peptide. So this is nevirapine with HLA-DR 0101, DR1. So what about carbamazepine? So and Xuan Yu just gave a great talk, and this fits. I'm so glad that we just saw your data, because what I'm going to do now is try and present data that's consistent with yours. So does the drug bind under the groove like abacavir? If it did, you would expect it to alter the repertoire of peptides bound to the HLA molecule. And so far, that has not been the case. As I understand, 3 different groups around the world have done that experiment, and there's no evidence that the drug carbamazepine alters the repertoire of peptides that are bound to HLA-B1502. Not only that, the modeling suggests that that probably doesn't happen, because if the drug binds that site without peptide, then it would prevent the peptide from assuming the standard conformation that peptides take when they bind class 1 MHC. So it seems unlikely to me that the drug binds under the groove, under the peptide like abacavir. Unlikely to be the repertoire altering idea. Can the drug bind the HLA and the T-cell receptor outside the antigen binding the cleft, acting as a superantigen, where the CDRs would play less of a role? They would be less important if the drug bound over here. This also seems unlikely, based in large part on your experimental data, which shows that the CDR usage actually is important, especially CDR3s. And the molecular docking is consistent with this result, because if we try to dock the molecule to these surfaces, the predicted binding interactions, the predictions are weak binding, weak. So probably not the case. So is it possible that the drug might bind as a conventional antigen, where it would be interacting with both the HLA molecule and the T-cell receptor complementarity determining regions? This would be consistent with the data that we just saw. And these are, remember, these are the sequences from the alpha and beta chains that were identified in Xuan Yu's studies. So this is the experiment that we did. In the couple of remaining minutes, I'll mention this. So we basically modeled B1502 based on the crystal structure of B1501. We modeled a peptide, and the peptide that we modeled is derived from the sequence motif that was identified from the Illing-Rossjohn-McCluskey paper, where they eluted peptides off of cells that were treated with carbamazepine. They derived a motif, and we found an endogenous peptide that corresponds to that motif, and that's what we modeled. Then we docked the drug on the HLA molecule with and without the peptide. We also docked the drug on this T-cell receptor, this V-alpha-12, V-beta-12 T-cell receptor that has the CDR3s that are most commonly used by the SGS-TEN patients. And in addition, we generated a model of a complex between this T-cell receptor and the peptide in MHC by superimposing these models on top of the solved crystal structure, one of the Rossjohn HLA-B structures. And then we asked, are there structural pockets at the interface between T-cell receptor, peptide, and MHC that are compatible with binding small molecule drugs? And we used a program called the F-pocket to evaluate these pockets. And then we docked the drug into all the pockets that were druggable. And the result, I'll just skip to it. The most likely answer is here. So it appears that the highest score, the best prediction that we arrived at after doing all the modeling that I just talked about, is supporting the idea that the drug might interact as a conventional recognition mechanism where the drug makes contact with both T-cell receptor and HLA, and the CDRs are used. So this is a top view looking down on the T-cell receptor. Here's V alpha, here's V beta. This is the alpha 1 helix of the HLA molecule. Here's the alpha 2 helix. Here's the modeled peptide. And in magenta here, this is the CDR3 from the alpha chain. In green is the CDR3 from the beta chain. In yellow is carbamazepine. And if we rotate the structure so that you could see it from a different angle, this is what it looks like. And what you can see is that the drug appears to be wedged in between CDR3 from the alpha chain and CDR2 from the beta chain. And I see my time is about up, and maybe next time I will get a chance to tell you about an exciting project that's being supported by the FDA, where we do the similar type of technology, trying to study which small molecules interact with different HLA molecules. And here what we've done is we've looked at about 49 chemicals that cause delayed type hypersensitivity, and we looked at the 28 most common HLA-A alleles and the 30 most common HLA-B alleles, and predicted binding with these chemicals, because ultimately we're trying to predict who is at risk for developing an adverse drug reaction. So why don't I stop here? Thank you.
Okay, so our final speaker for this session is Michael Rosenblum, from Assistant Professor of Dermatology at UCS School of Medicine. Thank you.
So, is it this? Okay, gotcha.
We just need it loaded. It's up there now.
This one. All right. I'd like to thank Elizabeth for inviting me. I'd also like to thank Jean and the Anderson family. Your testimony and hearing your story really is inspiring. For us that go to the lab every day and try to figure out these very difficult and complex diseases. So thank you very much for telling us your story, sharing your stories with us. So I'm an immunologist and a dermatologist. I specialize in seeing patients with inflammatory and autoimmune diseases of the skin. And in the lab, I study the pathogenesis of these diseases in sort of a functional way in my laboratory. And we used to think that the way that individuals developed these autoimmune diseases was that they just were unlucky. They had these T cells that left their thymus and were capable of reacting to and responding to tissue antigens, and that people that didn't develop these diseases just didn't have these T cells, and the ones that did develop these diseases have these T cells. We now know that that's not true. Actually, almost everybody has T cells floating around in their blood that are capable of reacting to and responding to self-antigens. And so, why then do some people develop diseases such as these inflammatory autoimmune diseases, and others don't? Well, we know now that these autoreactive cells are constantly being balanced by regulatory cells. And so the body has a mechanism to actually regulate these cells so that we all have these cells that can mount these autoreactive responses or autoimmune responses, but they're being balanced by regulatory cells. And really, it's a disruption. The paradigm shift in the field of immunology that's happened over the last 10 years is that it's really a disruption in this balance that results in autoimmunity. And that if we better understand the fundamental biology of how immune responses are regulated naturally by the body, that we could better understand the pathogenesis of these diseases, and it will help us develop new treatment strategies targeting these diseases, which I'm going to talk a little bit about today. So, well, by far and away, the most well-studied and widely accepted regulatory cell in the body is the regulatory T cell, or Treg. It's defined by the expression of the transcription factor FOXP3. It's a CD4 positive T cell. And these cells play an indispensable role in suppressing inflammation. They're derived in the thymus, and they're also can be derived in peripheral tissues. And these cells have the predilection to work and to regulate immune responses in the gut and in the skin. And mice that do not have functional Tregs die very quickly from fulminant autoimmune disease, and people that don't have functional regulatory T cells die very quickly from autoimmune disease, systemic autoimmune disease, suggesting that these cells are critically important in regulating inflammation in humans, evolutionarily conserved from mice to humans. Now, how do these cells work? Well, these cells work by a variety of mechanisms, and my mentor, who writes the immunology textbook, whenever students ask him how do Tregs work, he said the answer is always all of the above. So these cells have multiple mechanisms by which they can suppress immune responses. And so you can imagine that augmenting these cells is not just augmenting one pathway, but it's augmenting all of the pathways that they use to suppress inflammation. The most widely accepted mechanisms of Treg-mediated suppression is IL-10 secretion, expression of the inhibitory receptor CTLA-4, and IL-2 consumption via expression of the high-affinity IL-2 receptor CD25, and I'm going to talk a little bit more about that coming up. Suffice to say, these are highly potent suppressive cells that regulate and control inflammation in the human body. And so I want to talk, my lab studies these cells in detail, and we specifically study these cells in tissues. The skin is one of the main tissues that we study. And so we've done a lot of work looking at how these cells function in mouse skin. And just sort of for some characterization, these cells are highly enriched in mouse skin. This is just a FACS plot showing that in the lymph nodes, about 10% of the CD4s are regulatory T cells. But in the skin of mice, about 50 to 60% of these cells are regulatory T cells. So almost half of the CD4 compartment in mouse skin at any one time are these cells, these suppressor cells, or these regulatory cells. And we know now that these are tissue resident cells, and that's just quantified here. Back in 2011, we created a model of skin inflammation where we induced a neoantigen into the skin of mice. By giving these mice doxycycline, we were able to induce a neoantigen in the epidermis, and the T cells in these mice got very angry and upset, and they reacted to this neoantigen in the skin, resulting in a very fulminant skin autoimmune response characterized by sloughing of the skin and hair loss and weight loss and erythema. And so what was most interesting about this model, though, is that this disease actually spontaneously resolved over time. So despite the fact that we had this antigen, this neoantigen being expressed in the skin, that with time, the body just naturally actually resolved this reaction. And you can see that these are photos here of mice that, at the height of disease, showing fulminant skin inflammation. However, some mice that we keep on antigen for 60 days, so the longer period of time of continuous antigen expression, they spontaneously resolve and look like mice that had never actually been, that we'd never actually turned on antigen. And so these results really suggest and kind of confirm what we knew, is that the skin specifically has an intrinsic ability to regulate inflammation. And these are regulatory mechanisms that are kicked in after this fulminant autoimmune response is initiated. And so we thought that maybe regulatory T cells were involved in this because these are the major players in regulating immune responses in tissues. And so we did an experiment where we just basically depleted Tregs at the height of disease. And you can see that this is just a schematic. We turned antigen on in the skin. These mice developed this fulminant inflammatory skin response. And then we depleted these regulatory T cells at the height of disease with the idea that if these cells are involved in actually the resolution of disease, by depleting these cells, we wouldn't actually see this disease resolution. And that's exactly what we observed when we treated these mice with a Treg depleting antibody. The black line is shown here. They didn't resolve their disease, and actually many of these mice succumbed to the disease because it was so fulminant from systemic inflammation or sensible water losses. So this really suggests that the Tregs specifically are the main cells that are responsible for regulating this autoimmune response. And if you don't have these cells, actually you don't resolve this reaction, and it becomes fulminant and actually lethal. So how about human skin? So we also have extensively studied these cells in human skin, and it turns out very similar findings in that in human skin there's a large percentage of Tregs, this is shown here in blood and in skin, and this is actually quantified here, that the skin actually is a huge reservoir for regulatory T cells in the human body, and there's actually 10 times more Tregs in skin at any one time than there is in the blood. Probably the biggest reservoir of Tregs in the human body is the gut, and then the skin, and then probably the thymus. So these cells are highly prevalent in human skin as well. And we've done a lot of work defining these cells actually in human skin. So naturally, if these cells are such potent immune suppressors, and this is actually the way the body naturally regulates immune responses, then pharmacologically augmenting this cell population might actually really have clinical benefit. And this is the new way of thinking about autoimmune disease and treating autoimmune disease. It's not by targeting specific pathways on the effector or the pathogenic side, but it's actually bringing up the regulation and enhancing the body's own natural ability to suppress these responses. And so this is from a figure that we published a couple years ago showing ways that people are trying to augment Tregs for clinical benefit. One is by taking these cells out of the blood, growing them up into high numbers, and activating them ex vivo and giving them back to patients. We have 2 studies ongoing at UCSF doing this in cutaneous lupus and pemphigus. And we also have studies in type 1 diabetes as well as transplant recipients. This is a very time-consuming, expensive process that requires 3 to 4 weeks to grow these cells up and give them back. Another modality that I'm not going to talk about is actually giving antigen-specific peptide. In the absence of adjuvant, you can augment Tregs that way in an antigen-specific fashion. What I really want to talk about is IL-2 treatment, low-dose IL-2 treatment. Tregs were initially defined by their high expression of the high-affinity IL-2 receptor. And these are the only cells in the body that really express really high levels of this IL-2 receptor in the steady state. And so people have started to take advantage of that by giving patients with autoimmune disease IL-2 or low-dose IL-2 to preferentially augment Tregs and again, restore that immune balance or that enhance that regulation. So this is a study that was just only a 5-patient study in JAMA Dermatology in 2014, which take patients with alopecia areata and gave them low-dose IL-2 therapy. And we can talk about the difference between high dose and low dose. High dose is actually used in cancer and it activates a lot of effector cells. Low dose is actually used for autoimmunity because it actually preferentially augments Tregs. And they found that these patients grew their hair back, suppressing the autoimmune response associated with the hair follicle in alopecia areata patients. A little bit more press and a little bit more rigorously studied was the effect of low-dose IL-2 in treating chronic graft-versus-host disease. So this is there was a published paper in 2011 by Jerry Ritz's group in the New England Journal of Medicine showing that if you augment Tregs, you can actually treat chronic graft-versus-host disease, a very difficult to treat autoimmune chronic inflammatory condition. There's been subsequent studies by that group since then that's shown that if you can give patients low-dose IL-2, this is specifically augmenting the Tregs. This is the Treg to T conventional ratio, that balance that I showed you before. They're increasing the balance in the form of regulation, and these patients are resolving their disease. Again, a very difficult to treat autoimmune condition. And then more recently, in a paper published in Nature Medicine late last year was looking at the effect of low-dose IL-2 therapy in the treatment of lupus. And so these patients have systemic lupus, and they were given low-dose IL-2 therapy. They preferentially augmented Tregs by giving the low-dose IL-2 therapy. And what they were able to see is a really nice clinical response in systemic lupus. where they actually had, these are just lupus clinical responses, and they're lessened the dependency on systemic corticosteroids, and it was quite a pronounced effect in this cohort of patients with treating with low-dose IL-2 therapy. So what these studies show is this is the tip of the iceberg, and I think this is the future. This is actually, using a new way of thinking about the immune system, and that augmenting the regulation works and actually has been started to use clinically to treat conditions. And I guess autoimmune and chronic inflammatory diseases, and I leave you with the question, could this be done for TEN, SJS, TEN as well? I mean, could we give a drug that specifically augmented the regulatory compartment, the regulatory cells, in a way to quickly reset that balance between effector T-cell responses and regulating those responses? And so obviously it hasn't been tried yet in TEN, SJS, but I think it's a plausible treatment option. And with that, I'd just like to thank members of my lab and funding sources.
So that opens this session up for discussion.
Michael,
Yeah.
what happened with the T supercell cells in SGSTN? They are activated, they are not, they don't have time to suppress?
It's understudied. So I went and did a quick lit search on Tregs and SJSTN, and there's really nothing out there. Those studies have to be done in a pretty sophisticated way, because if you just do immunohistochemistry of tissues, it's not very good, because you only can look at CD4 and FOXP3, and those two markers alone are not good enough to say if it's a Treg or not. I would argue that patients that develop T and SJS do have a default in regulation, and that might be actually a predisposing factor where you actually need that sort of genetic or default hit in your ability to control immune responses, and then once you get the right antigen and you have the T cells and you can't regulate it, you get the disease.
Just a brief comment on the paper you showed from Nature Medicine on lupus. In that study in China, it was uncontrolled, and they also had the patients on prednisolone. So there's considerable doubt about that. Mm-hmm. And its use in such an acute condition such as SJS is a much higher bar, I guess. So it just seemed, do you think it's more of a challenge in terms of how much inflammation you have to turn off and how quickly? Because we don't know till we look, but...
Yeah, so, I mean, we don't know until we do it, right? But I would argue that, even if you do... So these are potent regulators, right? And so in every mouse model, they've shown to actually attenuate disease or reverse disease. And that's mice. And so I get the caveats. I would say that even if you do activate some of the effector cells in autoimmune diseases, whether it be chronic or acute, you're going to hit every Treg. And so that actually should shift the balance in terms of regulating. So I would be less concerned about activating effectors. I'd be more concerned on the on-target effect, that you're actually getting a good enough dose to activate your Tregs.
So also for Dr. Rosenblum, I was part of the GVH trial in Boston. What we find is that after a while, we do lose response to IL-2 in GVH. Okay. For other autoimmune diseases, how well does the IL-2 receptor modulate up and down over chronic exposure and flooding of the receptor, so that you may actually explain loss of that, is question number one. Mm-hmm. Then when we looked at IL-2 expanded Tregs and reinfusion into GVHD, the response was delayed further, but also lost. Mm-hmm. Then when we added photophoresis, which increases Tregs, you keep on doing the same. So how worried are you about escape mechanisms in this, and can you modulate that? Those are two questions in one, I realize.
They're great questions. They're great questions. I mean, IL-2 turns on its own receptor, right? As you know, right? So the more IL-2 you give, the more you should get expression of the high affinity receptor. So I'm unaware of receptor down regulation in the context of chronic IL-2. I don't know the answer to your first question. I do know myself and others have been involved in actually developing drugs that don't hit the low affinity receptor and only hit the high affinity receptor, the so-called IL-2 muteins. Yeah. And I think these might actually have different pharmacokinetics in vivo. One thing that I will say about about actually the loss of response is that soluble CD25 probably goes up over time, and then that might potentially be neutralizing. I don't know if you guys have looked at it at all, but that's one concern, right, is that if you keep increasing your soluble CD25 levels, because IL-2 will turn on CD25 expression, it can get cleaved and then become soluble. If you give the same amount of IL-2 at that point, you might just be binding the soluble receptor and not really engaging the target on the cells. That's hand-waving, but a possible explanation.
And one other question. Have you seen the same result when you use IL-2 for autoimmunity in children, where presumably thymic function is clearly preserved?
Yeah, we haven't done any of that in kids as of yet. Yeah, we haven't done any of that in kids.
Thank you.
Yeah.
Hi, I have a question. Probably most of you could answer this, but I'm wondering about these proposed hypotheses of HLA links with the different peptides, and whether you're suggesting that they're activating the T cells independently of co-stimulation, or whether you think co-stimulation is necessary. And I wonder because when you're only seeing 2% or 6% of a population with an HLA responding, if you think that they have to have some other hit like an infection at the same time, to get that sort of secondary signal, you need to stimulate your T cells. And also, if perhaps that is why we often see patients who actually have URIs, or what we think are URIs, and then they get the drug and then develop disease. Anybody?
Excellent question.
Or no,
I bet we all have our opinions. That's a great question. The degree to which an infection might be associated with a particular drug, my guess, based on the literature and the experts in the field, many of which are in this room, is that it may well be significantly different depending on the drug. The mechanism for how the drug might interact with the peptide might be different. The infectious agent that might be involved might be different. So there are a lot of unknowns, but there are some commonalities that we have seen in this meeting today.
Has anybody ever taken, I suppose APCs, I should say, out that have already taken up antigen and maybe cultured them with T cells in the absence of any sort of other co-stimulation? Has anybody ever seen T cell proliferation in that setting, or has it always been done in very in vitro-like?
Well, when you say co-stimulation, I guess most immunologists view that as involving the so-called signal 2, meaning CD28, CTLA4, PD1.
Right.
I'm not sure if that's what you're referring to, or if you're referring to a co-stimulation like another stimulant like an infection.
Anything. Anything, yeah.
I know that Wen Hung has done a lot of experiments with treating antigen-presenting cells, taking the drug away, and then trying to stimulate the T cells, and trying to evaluate whether antigen processing is involved or not.
Yeah, we have done several experiments to realize whether the peptide, so-called peptide or viral peptide, adding to the drug antigen. So unfortunately, that result didn't show a very strong result. Maybe you can explain. We hope to find the viral top that can add on effect to the T cell activation when combined with the cabozanthine or RP or oxcabazepine. The result is not very solid, but do we identify some viral peptides that can have potential the binding with the drug antigen can provide a better the antigen response to the T cell. But it's still, the data is not very solid. Maybe you can explain too.
Regarding the special peptide, I think the special peptide can be possible only in the cabozanthine-induced SJS and TN, because we only find the special T cell receptor in this kind of drug-induced SJS and TN. And but we cannot find an oligoclone, cannot find a public T cell receptor in other kind of drug-induced SJ and TN. And another question is regarding the signal two of the SJS and TN. We ever culture the the PBMC of the patient with the drug and with the anti-PD-1 and anti-CTLA-4. We find that if we culture the PBMC with the checkpoint inhibitor, then we can find a higher response rate. So I think because the drug is a difficult antigen, so I think this method can help the adult TTAs.
And that's actually the point I was going to make, that what you did in those studies is actually decrease regulation by binding CTLA-4 and PD-1. So if there is a defect in immune regulation that's predisposing, you actually need a lot less co-stimulation.
Thank you.
We might just have
a short comment to Professor Abe. I think your theory is appealing, that the Helicobacter pylori, its antigens themselves could be responsible for the rash. That reminds me of 30, 40 years ago, a similar theory has been proposed for explaining a rash at the beginning of imidazole treatment in patients with candidosis or occasionally other mycosis. I don't remember a precise bibliography, but you have found that, or it may be worthwhile because the hypothesis is basically the same. And I had a question to David Ostroff about one of your slides. I am not able to remember one, was measuring some index of linking of several drugs to HLA B1502. And what my impression for, it was far away and slow characters, was that the binding effect was something like 9 for carbamazepine, but it was also the same range for all other anticonvulsants. Phenytoin, is that correct?
Yes.
So then what is the specificity? For me, the linkage of 1502 and carbamazepine was rather specific in terms of ability to induce the disease. And if you think that an essential point is the linkage to HLA molecule 1502, how do you explain that phenytoin and also, don't remember, but maybe phenytoin, yes, and maybe also another anticonvulsant, don't remember. What is your explanation?
The short answer is that the drugs that have 3 rings that are linked to each other, they may bind in a manner that where the peptide has to be there. In other words, the prediction without the peptide, with the high number, that remains to be validated. I am reluctant to believe that prediction in the absence of experimental data. Instead, my guess is that for phenytoin and the other drugs that have 3 rings that are linked to each other, the situation is going to be more like what we saw for carbamazepine.
I think we might just leave it there.